this for a long time. It basically is part of this. So again, I probably have more time to do anything on Thursdays than any other night or any other day. But effectively, when I do muscles, I send that all to you separately. I'll go over it. We may have a time to do a little bit of it in lab. I would rather, the reason I want to get this started is so you can dissect the brain, preferably hopefully on Monday. We'll be able to, even with just doing a little work on it, because I know we have the exam on Monday. And so this is sort of the introduction to the brain, and I mean, I have to show it, and I'll take a picture, and whoever, that's another Ava, right? Yeah, it's over there. Can't tell that. Now I begin to know most. So, yeah. Effectively, the construct of the human brain starts with the highest level called the cerebrum. C-R-U-M. And effectively, in the cerebrum, after that, we get to something called the basal nuclei. These two are the highest functioning areas. And as you work your way down, it's like here now, go down to the bottom, you have a relay center. So what happens in the middle is interesting. After this, you get to a level that somebody might describe as almost subconscious. Okay, it's called the diencephalon. That name kind of, and there's different components of it. The diencephalon has the thalamus, the hypothalamus, and the epithalamus. Associated with this is another area that is really not the same as these, but it's sort of in the associated area. It's called the limbic system. Limbic system, it sort of encompasses several areas, these top three functioning areas. So the next area, and the next lower order functioning area is called the midbrain. Sometimes with the old term for it, as you'll see shortly, the mesencephalon. And now as we begin to get farther down here, starting with that midbrain or mesencephalon, M-E-S-E-N-C-E-P-H-A-L-O-N, handwriting you're going to see in a minute. You have, this is the start of the brain stem. And after this is something called the pons, and then the medulla oblongata. And here, this is really, it's not involved in any conscious activity. So this is referred to as the brain stem. And then finally it gets its spinal cord. That's the transition. So here, we're going to, and I'm going to go over each of these areas and sort of give you a heads up of what's going on. Interestingly enough, and I probably should have done it without the screen, something that effectively sort of involves all of these areas to a lesser extent and relays signals. I don't have a place to write it. So important. It's called the cerebellum. That's probably the second highest order functioning area, even though effectively it's not, we're not consciously aware. It's a very, very interesting relay center that picks signals up coming from below, picks signals, sends them, relays into the appropriate area, and basically incorporates them. So the cerebellum is a really significant area that involves all of it. Whether I get to explain it fully today, I do not know. I've been sort of doing this presentation since I started teaching. You know, more or less every year for the last, I don't know, almost 30 years. Jeez, I'm old. So the whole idea of how this works, yes, sir. I emailed them all to you. Didn't I? I thought I was on my game. They're too big to fit in this whole ultra. I mean, the only ultra I want is a Michelob Ultra because it's low carb. I kind of, if I drink, I really like the Yingling Flight better. Not that you guys are familiar with any of those items. Moving on. So the whole idea of the development of the brain. What is the brain? Okay. If you want to think of it, I don't know how to present it to you in a particularly clear way. Use piece of paper. It's here. Effectively, this goes back to your really your elementary zoology. Those neat things that make a vertebrate. Okay, dorsal hollow nerve cord, right, notochord, all that good stuff. Basically, it's a neural tube. It's a tube that has basically, as it thickens, the exterior of this tube effectively becomes your nervous system. The interior is a fluid-filled area. Okay, so effectively, it's a neural tube, and some of the other structures will be incorporated in the notochord. We already, the notochord you already looked at. That's that nucleus pulposus that was in the spinal cord when we looked at the bones and the vertebrae. The pharyngeal pouches in us are not, we don't have gill slits over there, the middle ear area, that's the pharyngeal pouches. And this becomes the brain. It's filled with fluid, and what does it do? It expands, and it begins, like everything else, folding over on itself. That folding and that expanding is what you're going to get a little bit of familiarity with, hopefully in this presentation. So it's called cephalization. Basically, the top is the highest functioning, so to speak, going to the top. And other than that, so they call it the rostral. And eventually it becomes anterior portion of the brain, because as it's folded, it sort of pushes its way forward. All of which is not extraordinarily important of how it gets there, but this is the highest level. It has the greatest number of neurons. Interestingly enough, that area in the cerebellum has the greatest number of synapses, which are nerve-to-nerve connections. It's very interesting. This area of the brain that you will see when we talk about the cerebrum, and the cerebral cortex is only the outermost portion of it. When we talk about the cerebrum, including the cortex, that's about 80% of what we call our brains by volume. And about another 10 or 11% is that cerebellum. The rest of the stuff, the brainstem and those other things that I wrote up here, thalamus, diencephalon, blah, 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 not so much. Not so much. So here, the brain and spinal cord, here's the neural tube. Eventually, they form into these expansions and constrictions and folds, all of those things. And eventually, what we describe as this higher order functioning is effectively the forebrain. And you'll see that involves the cerebrum, the basal nuclei. Years ago, they were called basal ganglia. You'll sometimes see that. I erroneously refer to it that way. And you also the diencephalon. They are the highest order functioning, the forebrain. The midbrain maintained its name. Encephalon, the encephalitis is inflammation of the brain. Hence the encephalon, implies brain. Mes means middle. So the midbrain retained its name. And it's important when you understand some of the passageways that are involved. And the hindbrain is really the pons and medulla. This and the midbrain or the brain stem. When they're damaged, it's big trouble. When there are tumors there, it's big trouble. Someone has a stroke there, it's big trouble. What I'm saying is they're most likely not going to survive, because all what we call autonomic functions, the vast number of our autonomic or automatic functions are controlled there. You lose those controls, you can't control heart rate, respiration, blood pressure, et cetera, et cetera. Bad news. So, and then they talk about the secondary vesicles, the telencephalon, again, the cerebrum or cerebral lobes. Now we're going to start to see the bilateral symmetrical, basal nuclei or ganglia, which are sort of islands of what's called gray matter buried in the diencephalon itself. The midbrain stays. These divide. And you don't have to know metan or myelencephalon. I try to stay away from those terms. I mean, that's classic neuroanatomy. If you're going into if you're going to do a graduate program involving neuroanatomy, you're going to learn those. So the telencephalon, the cerebral hemispheres, okay, making up the cerebrum. Here's what I wrote down before. I didn't include the retina. The retina is really the sensory organ for which you see, And what comes backwards from the retina, which obviously is in our eyes, is the optic nerve. And all of those signals, as nearly every sensory signal that you have, shuttles to a thing called the thalamus, and it's a relay center. It basically, okay, vision, you're going to the occipital lobe. Hearing, you're going to go to the temporal lobe. Okay, general sensation, you're going to go up to parietal. So it's directing, it's a traffic director. It sorts, edits, sends its on. See, I'm trying to give you my concept of how it works. So maybe it helps you understand how it works and makes sense of the, even though, and from our standpoint, test-wise, because Jenna will certainly ask the question. Okay, yeah, the physiology that's going to be on, you have to know a little nugget about that. Yeah, highest order functioning is going to be the frontal lobe. Language skills, mathematics, reasoning, test taking. We'll see how well your frontal lobes were working the other day, if at all. One freaking smile out of the whole place. You're excused. No, she's excused behind you. I don't like you. Fox Chapel girl. I gotta be nice to her. Moving on. And then all of that cavity I was talking about, that's buried in here, you're going to see what are called ventricles. They are filled with fluid, and they're so important because A, they're filled with fluid and they provide buoyancy. B, that's really where spinal fluid produces the wrong term, finalized. Spinal fluid is a little bit like it's a little bit like plasma with some different stuff in it, and there's areas within these what are called ventricles. So these are ventricles that are very unique. We get to the heart has ventricles also. They're filled with blood. So I think they derive it. Here's what it looks like. So here's the tube. That area is all neural tissue, brain material. Look at the expansions. Now look at it beginning to fold as we get here. When you begin to see gray matter, there's a consistent pattern. Okay? Consistent pattern that you have, just to look at it globally like this, you always, in the center of the nervous system, you have fluid. Obviously, CSF or cerebrospinal. Surrounding that, then you have what's called white matter, connections that go up and down. When you get higher order functioning here, in the highest order functioning, you get gray matter, which is really nerve cells and their interconnections. White matter implies myelin sheaths. I'm sure you talked about myelin previously in other courses. That's that sort of protein, fatty stuff that insulates nerve. It's basically for conducting impulses rapidly. Myelin, the more myelin, the faster it is. The thicker the nerve, the more myelin, the faster. And so this becomes what we call cortex. In certain areas, that's a basic construct. In certain areas, you're going to see what we call islands of gray matter. And they are usually indicative of certain functions that occur. Just to give you a general construct, so as you're seeing it here, for instance, in the telencephalon, this is going to become the cerebral hemispheres, and the cortices are out here. That's the cerebral cortex. It's remarkably smaller than you think, but that's really where the ultimate processing, your ability to answer a test question, your ability to communicate, to reason, to solve problems, ways that we interact, all of that, basically the interpretation of material and assimilating of it, grasping it, retrieving it from multiple areas for memory, all comes there, okay, from those areas. And again, that's where we are identifying the signals that we recognize as sight. We are identifying the signals. I mean, basically, we're constructing them, identifying them, shuttling it to that frontal lobe and saying, this was the image I saw. Oh, that's a cat. Or, oh, that's a dog. Or, oh, that's a fumble or an interception. Whatever. Pick whatever example works for you, whatever your favorite thing to look at is. Okay. So all of that's part. Notice another area down here. That's the origin, as you can see, of the cerebellum. And so it implies when we have external gray matter, it implies high order of functioning. We'll get to the islands of gray matter. Notice here the diencephalon, thalamus, hypothalamus, epithalamus, these outpouchings are going to become eventually the optic nerve and what's called the optic chiasm, something you're going to see. And that's why I want to get into talking about this so we can dissect the brain, preferably on Monday, and have a little fun with that. Because I'll be able to bring some of this stuff in the material I will send with you, along with the review recording I know, Jeff. You're allowed to nag. It's okay. Send emails. I didn't get it yet. What the F are you doing? Sorry. My wife will realize it's a student. They talk to me like, did I ever tell you how I met my wife? She was my student. Don't judge me. It's okay. There's not that big of an age differential. It's not quite 7 years. I'm older. Not there's anything wrong with that. 20 years ago. You're judging me. You give me the stink eye. She'll know. She has such a nice way of speaking to me, because your study guides suck. You know, that kind of stuff. Don't hold back when you get old. Moving on. So you can see it's the same construct I gave you. Now, the fluid-filled areas in each of these constructs, so to speak, have different names. You've got to know these names anatomically. This is one of those things you're going to have to label for the lab. You have to understand from a test perspective. So we name the ventricles. Years ago, I'll show it. It's better for me to show it to you. Forebrain, this is just how it develops, doubles back on itself, envelops, blah, blah, blah. Creasing and folding. Interesting stuff. And I'll get to those fluid things in a minute. This is what it looks like week 5. Gestation. Yeah. Look, everybody. This is me. You're going to get my perception about and how I present it to you. I don't want to be controversial, but the brain's already developing at week 5. That's at week 13, end of the first trimester. That's a brain. You have the start. All the equipment is there. You can see the cerebral lobes. The midbrain is forming. This is the thalamus, the cerebellum. I mean, if you take a look at those are all the names. This is the brain at birth. And you flip back to that, I realize it has a lot of developing to do. It's there. But that's as far as I'm going to go with that kind of a discussion, because it's really not. It's a science class. It's not me talking about what I think certain things should be or not be. Okay? So this is what it looks like, and it's what it looks like in you and I. And to a lesser extent, only because it's smaller, when you see the sheep, it will look like that. It's just because it's smaller, and it doesn't have, these are called convolutions. Gyri that pouch out, and sulci are these little indentations. The sheep doesn't have as many. There's just not as much. The sheep would be teaching the course. The sheep would be dissecting us if the sheep was smart. Right? The sheep aren't that brilliant. They might be brighter than my dog. That's not gifted. Nevertheless, here it is. You're going to see this. This is the pituitary gland. It hangs down from the thalamus. It's an intricate layer. And I'll try to go into all of these things a little bit, ever so briefly in the time. But I do want to show you this. Gray matter, non-myelinated nerves, cell bodies. We don't use the term nucleus. We use a lot of what I would call archaic terminology when you're in neuroanatomy, because it was really the first system to discuss. So the term nucleus was used for clusters of gray matter that they saw when they cut apart a brain. And they said, that's the red nucleus, or that's whatever nucleus. Okay? So they use a different term on a cellular basis for nerve cells. They call them cell bodies. Soma is another word. Perikaryon. There's a lot of different terminology. Nucleus is not the appropriate. They use terms like rostral and caudal. toward the head or toward the tail, cephalic or caudal, rather than superior inferior. They use dorsal and ventral, back and belly, rather than anterior and posterior. So, I mean, it's not that I want you to be aware of it. I'm not too judgmental if you would, when it should be saying dorsal something, and you said posterior something, I'll buy it, because I know you're trying to you got the message to me. But going forward, that may not work, depending on where you're heading career-wise, educational-wise. So here's the white matter is mostly myelinated. The non-myelinated are somewhat hitting. Here's the basic pattern: central cavity surrounded by gray matter with white matter external. So you'll have little islands of gray matter, as you will see around it, particularly with the spinal cord, you'll see it. So I should have been a little more distinctive with that. And again, you're going to have little bits of gray, and then with your higher function, you get more of these islands of gray. So fluid, gray, white. Spinal cord, white's on the outside. Really, there's no additional processing. It's signal up and signal down. So voila. Here you can see this is spinal cord, way down here. Classic pattern we'll go into when we do CNS, and this is part of when we do the CNS, the anatomy of the spinal cord. Small central canal, this butterfly-like pattern, letter H, whatever you want to call it. And we'll name all the, these are called, white is called columns, gray is called horns. You'll get there. And so we work our way up. Next area. So this is a different area. So now we're coming up here. We're in the brainstem itself. This is the medulla oblongata. Right below it is the spinal cord. So you have a rather large cavity, and this is called the fourth ventricle. You'll see it in a second. You have some gray matter around it internally, and then you have these islands. Okay? These are called olives. This is mostly white matter mounds. They're called pyramids because they were sort of pyramidal to people who were looking at them. They also have a variety of cranial nerves that we will study and go over. The 9th, 10th, 11th, and 12th cranial nerves, plus parts of the 7th and 8th, all kind of start in here. And we'll go over why that is so significant in that area. And now these are hollow cavities that you will see why we make them. Why I'm making such a big deal out of this. This is a typical cut. When I did at Chatham for the Doctor of Physical Therapy and the Masters in Occupational Therapy students, I did their graduate neuroanatomy. I was doing the lab, someone else was doing the lecture. And it's a type of thing where they'd have to identify where the slice was. That's the degree of difficulty. You certainly won't have to do anything like that. But it's based on these ventricles. That's when you see MRIs of the skull and CTs of the skull. There's anatomical things. If you see eyeballs, you kind of know you're at the level of the eye. Just for instance, here you know you're here and you're going to see why. But this is part of the lateral ventricle. Here you can see these are going to be called the nuclei. You see the external gray, that's the big clue. And obviously, if you look at it, you have a little bit here. This is called the temporal lobe. And you see a little bit of the frontal lobe here. And look at the substantial. There's much more white than you think. This is where they are. We'll get to the cells. The cells are something we do in physiology. They're part of nerves themselves. When they're destroyed, they will not repair effectively. They just form scar tissue. Certain areas there's repair in long tails in the axons of the nerves. That's again more functional than true anatomy. The ependymal cells are part of this group of cells that support. They have their own connective tissue cells. Okay, unlike the connective tissue cells we talked about previously, we're talking about loose areolar and dense irregular and the different kinds of lining cells, all that simple squamous, all that crap. Okay, the ependymal cells, nerve glue is what neuroglia means. The glials, and oftentimes we call them glial cells. When you hear about a tumor, they'll call it a glioma. Sometimes very nonspecific and not a very good diagnosis. So the ependymal cells are in those hollow fluid chambers over capillaries that are sort of hanging down. That's where the spinal fluid is sort of finalized. And so you're going to see what they look like. And just to give you a little more anatomy, these large lateral ventricles you're going to see shortly are separated by a membrane called the septum pellucidum. So even though they are both filled with fluid, a membrane has developed between them. I had a student, a brilliant student, and she has one of her son, because I was teaching at BC3, so you get a lot of, that's where I met my one, return to, their non-traditional students coming back to school learning. And she had a son with missing a septum pellucidum, which caused more behavioral issues than physical issues. And so I'll show it to you first. You see these all the time. These you'll have to label, fair warning. This is what they look like. If you're looking at them, if I'm looking at the person, that's what, imagine that slice that you saw before. You have that C shape, sort of wider anteriorly, extending all the way posteriorly along this, the parietal area, all the way with a little mini tail in the occipital area. Okay, and then coming forward in the temporal area. These are all the major lobes: frontal, parietal, temporal. I think frontal, parietal, occipital, temporal. Notice there's a little opening there. That's a passageway called the interventricular foramen. It's an opening, not in bone, but an opening between these two, and it feeds the third ventricle that surrounds the thalamus. And yet there's another opening, which you'll actually see on the dissection. You'll see the ventricles on the dissection, the lateral ventricle, the third ventricle. You see the hollow areas. They won't have the fluid in them. In between the third and the fourth ventricle, which is quite large and really important, that sits, here's the cerebellum, that sits between the brainstem and the cerebellum. And so it's quite important because this is where cerebrospinal fluid isn't just inside, it's outside. Your brain's neat. What keeps it be able to float is you've got it floating on fluid on the inside and on the outside. The way it escapes to the outside, it's made in here, third and both lateral ventricles, the fourth ventricle. But this is the escape. It's got little openings called apertures. An aperture is an opening. Lateral apertures, one on each side, bang, bang. One medially sort of going, and that's how the fluid surrounds. And we talk about in this chapter the meningeal layers, surrounding soft tissue, protective around the brain. So that's why these are really, it really is how we separate them. And again, that view, that tells you right where you are. If you move it backward, it changes. And radiologists, in the advent of MRIs and CTs, I mean, you're getting continuous slices of these things. In the old days, we would have, rather than having like a video of it, we just put x-rays up on the wall and said you'd have a pack of films you could hardly carry, or a film that was giant and had all these slices, and you sort of had to put the puzzle together this way. Now, now you can see them almost in real time as things are flowing through, accelerate them, slow them down. I mean, technology has helped in that. How about the injury to Hill on Miami? Oh, we're talking about sports injury. That's nasty. Tyreek Hill. I mean, apparently he caught that. The turf was bad, beat up from University of Miami and from Dolphins, and he caught that, and between his foot couldn't move while his foot was being twisted. It wasn't a dirty play. It was just the trouble. In the old days, we used to get feet stuck in the artificial turf here at the different stadium here at Three Rivers Veterans. Sir.
Better to have that injury than play for the Dolphins, right?
Pardon me?
Better to have that injury than play for the Dolphins, right?
The way the Dolphins are, it's going to cost them a lot of money. The guy, Pro Football Talk this morning, they were talking about that specifically. I am not a fish fan. Okay. We don't play them this year, do we? Do we? We do. Yeah. He's not coming back. I'm not so sure he's going to come back at all. I mean, his age and Drew Rosenhaus was always paint the picture of optimism. Money. So here are the ventricles, the ependymal cells, the openings that I talked about were there. You can read through that. Same illustration of the illustration. So the hemispheres, almost, if I understand, 5/6 of the brain. Okay, gyri, outpouching, sulci, the grooves. Some of them are deeper than others, as you will see. Anatomically, there's something called the longitudinal fissure, the sagittal plane. So why I railed on that sagittal plane is this: is the divide between the hemispheres, and it goes along, dividing right and left. There's a fissure. Pardon me. And when you dissect the brain, you're going to go right down that fissure and cut through a rather substantial amount of white matter and demonstrate something you're going to see a lot of in these illustrations called the hemisection. It's like you took the brain, you could open it up like you would a book. It's there. You have a transverse fissure in the back. You'll actually see that. Okay? And that's between this right. Here's the longitudinal fissure. The transverse fissure you don't see here is between the cerebral lobes and the cerebellum. It's here. You'll actually see that. You're going to see the thickness because I order specimens I order, even though we won't get to dura, the dura monitor have the dura on them, the very thick, and it's substantially thick. I used to have a human brain when I was at Chatham doing that course. The professor was visiting from Pitt, and he gives me this. I went, could you come to my car and get the brain? I'm like, sure. And in this giant bucket, this white bucket with, just horrible. In the old days, with formaldehyde, it would be a hazmat right away. I had to have a hazmat license to carry. My son does. And we had a brain, we had spinal cord. It's crazy. And because I was teaching anatomy at a couple of community colleges, I took it with me. I mean, it's okay. I said, can I show it to my? He goes, yeah, sure, go ahead. Because it wasn't Chatham's property. It was Pitt's. And so here's my daughter with her buds outside. And she was like a rather raucous teenager. One day she'd go to me, Dad, is there a brain in your trunk? She goes, is that a human brain? I said, yeah. She goes, all right. Don't have children. Here, this is called the central sulcus. This is the divide between the frontal lobe and the parietal lobe. You won't see it on the sheep dissection. It's not distinct. Okay. The difference why it's a sulcus rather than a fissure. Sulcus is just more of an indentation. The fissure is relatively deep. Okay, and you won't see this. This is called the calcarine sulcus. That really is a division between the parietal lobe and the occipital lobe. This lateral sulcus you will see on the sheep. That's and it's lower than it is here in us. This is the temporal lobe, and a really important lobe, particularly for sound and voice processing, other sensations, big player in seizure disorders. And here's the cerebellum. And when you see sulci and gyri, again, that's indicative of higher order functioning. You'll notice the pattern is significantly different here than here. So here are the lobes, frontal, parietal, and this is like a little nugget that you have to know. So frontal, higher order thinking, there, and these lobes back. We have frontal, temporal, parietal, occipital. Frontal lobe, we have highest order, they'll give you a little. Behind temporal, mostly auditory processing. Occipital, visual. A lot of our circuitry, more than half of our really processing is designed now for visual. Very visually. And understand the human brain is extraordinarily fluid. Someone who loses their sight, their hearing, the circuits become dedicated, their hearing becomes very acute. You ever seen that, like, the Marvel show Daredevil? You gotta watch that, the particular one, and it's very cool. And it's amazing how losing one sight will amplify all of your senses. That's just the nature of the quote. It's great names called plasticity of the human. It's very important, believe me to me, and I'll have some illustrations from the weekend. Parietal's general sensation. So, that, whatever, that thing you tripped on this morning in the bedroom, that sticky stuff, I was in the hall and I go over your way, that's general sensation.
that.
Right. Basal nuclei, when your brain issues a command, it says, grab this. Well, how do I grab it? Do I grab it gently? Do I grab it forcibly? Do I push this way? What happens formally basically is just a command. How you execute it, whether you that, whether you grab it forcefully, has a lot to do with the basal nuclei. So imagine people who can't either shake on the way getting here, or can't pull it, or have difficulty, they have difficulty with movement disorders. This is typically the region with a variety of conditions that are not equivalent, they have the same name. Movement disorders. A lot of them are caused by a lot of medications that people take. Movement disorders are part of that. My first wife has to take a variety of medication. Unfortunately, some of the meds have resulted in some pretty severe Parkinson's. The insula, is a very, very primitive lobe. That's really where your taste and smell are very, very primitive. And that's part of the reason where that's the one part of your brain where your olfactory nerve, the first cranial nerve, goes by smelling salts work. It's the only nerve that goes directly into the cerebral lobes and activates something called the reticular activating system. It's what keeps you aroused or awake. And that's why you smelling salts to wake somebody up sometimes, for certain different things. So the insula is sort of buried, older. Here's a typical slice. In this book, red will be motor, blue will be sensory. Whether it's lighter blues or a darker blue, they're all implying sensory. That's, again, a fissure is quite deep, where a sulcus is not quite as deep. So we change the name as according to their depth that are there. The frontal lobe, for us, is the largest. Again, not so on the sheep. Unless it's a gifted sheep. I know, it's sheep jokes. Remember, did I tell you that? What one shepherd said to the other? Let's get the flock out of here. See, that's such an old joke. And everybody laughed at you. What's so interesting over there? It's okay. Nobody listens to me anyway. The insula is deep inside there. Central sulcus, those are going to be important. Pre and post central gyrus. When you initiate that command, that's called remember the central sulcus. So we say pre is the gyrus here, the mound here. Post is the mound here. When I actually generate the command, it comes from there. Post central is when I actually feel something. When I reached this and I felt a rough edge, I actually felt it by way of the post central gyrus. So and that's why the decision to grab my phone started maybe here and worked its way to here and finally to here. When I felt whatever I felt it here, and I began to process the information back here. So the areas of brain are very, very well understood in that regard, in, let's say, basic tasks or basic things. I mean, the whole why somebody's smart, why somebody's not, why someone's a savant in certain things. I mean, I can't tell you. Nobody can. I mean, I think neurologically and psychologically, there are people who know. So, again, here's the cerebral cortex, white matter inside, and deep are basal nuclei. They show you the same illustration. So here, cerebral cortex, awareness, sensory perception, initiating movement, relatively thin. What's that? That's probably not a quarter of an inch. Much thinner than you think for these high order functionings that are there. Cell bodies, dendrites, the axons are the white matter. Very heavy, 40% of the mass of the brain. Here's some of and this is how they determined, they use functional MRIs. So in the occipital lobe site, basically lit up with a different color. Hearing, temporal lobe, speaking, you're going to learn about this area. It's called Broca's. Proper name Broca''S. Very important. And thinking all over the place, obviously. A lot of it, in the context that I'll present it to you, is in terms of when it's damaged. We see a lot of compromises in certain areas. Something called a stroke. Stroke is blood, is brain tissue that dies from lack of oxygen. The tissue that dies the most easily, motor tissue. When a baby is born and doesn't have a sufficient amount of oxygen, okay, it's typically the motor lobes that are damaged first. Okay, and when the motor lobes, what happens afterwards, as the body starts to heal, that damage manifests itself in irregular signals going to wherever those muscles are. Classically, they're on one side or the other. If it's on both sides, it's very tragic. So if I have a stroke or you have a stroke, typically it'll be one-sided. Everything is contralateral in this context. We'll learn about some exceptions. So if I have a stroke on the left side, my brain, I end up with something called right spastic hemiplegia, half paralysis on the right, but it's spasm. Because that part of your brain basically sending extra signals to your muscles saying contract, contract. As you'll learn, flexors always overpower extensors. So we see flexion contractions. That's why people will have to wear specific braces holding their 90. They'll have what we call an AFO, an ankle foot orthosis, which holds their foot and ankle relationship at 90. Because other than that, it's going to start to contract under the drop foot. And once it adapts to a position, there's no way to fix it. It's always going to be there. And there's always going to be damage. It doesn't always mean it's tangible. But that's when I talk to you, and I miss very, very well. She's my grandkid. The little blonde girl, remember that one? She's a pediatric surgeon. Diagnosis she carries. Her left hand is immensely short. She can hardly reach. Right hand, right leg. She's playing soccer. She's very verbal. It's manifested. We're very fortunate. Doctors, what you do with patients is you paint the bad picture, and when it looks, you don't make promises you can't keep. I told my son and daughter-in-law, and this little girl was just 8. They said, "You'll be lucky if she can sit up." And I mean, now she's playing sports and all that type of stuff. 2nd grade, it's remarkable. So I mean, we are. It's a miracle every day that we begin to see it. But she does carry a diagnosis of cerebral palsy, which is what happens because of the damage, and not just her, but all of these kids. And there's more than you think. And they didn't realize it for about the first 6 months because babies aren't doing a whole lot of motor stuff. She had the head control. And it was more. He sent me pictures of the MRI and started asking me questions because I was kind of the first one to go, "Oh, how am I going to tell my kids what I'm seeing?" Because they're not medically oriented. So it's very, very interesting where this all plays. That's when, and part of that area in an adult, this area is commonly affected, and it's a very famous condition. We call it expressive, and the word is called aphasia, APHASIA. Someone, and when you try to rehab patients like this, they'll say, "Yeah, is it cloudy outside?" And they know it's not. They just can't verbalize it. And it's expressive, so it's not like, "Well, here, write it down." That doesn't work that much easier. So it's very, very frustrating to know the answer, not being able to say it. There's another area you're going to learn about back farther in hearing. That's called Wernicke's area. You'll see it. You don't have to write this down. Wernicke's is the opposite. We call it a receptive aphasia. In other words, and a patient knows they're asking you a question. They just have no idea what you're asking. So they might be saying to you, "What's your son's name?" "Oh, yes, it's a beautiful day." You get a perfectly well-constructed answer. That has nothing to do with the question. Because you're trying, you know somebody's talking to you, you're trying to kind of guess. The words don't make any sense. So we see a lot of these patterns consistently. You see a lot of interesting things over time here. that are there. And so motor areas, this is what's up in the street. We probably should make it time. I'll start here. I'll send you a discussion from here, and we'll go from here. Thank you, and I'll get the recording, and I'll get the grades done. All they do is nag.